Well, first of all, thank you very much for the invitation. It's an honor to be with you delivering this lecture and sharing some of our experience with you. Special thanks to Professor Alfredo Martinez. Organisms and their cells perceive the constantly changing availability of energy and nutrients and accordingly modify functioning of their genome. Until recently, it was generally thought that nutrients affected transcription in higher organisms, mainly by hormonal and neural mechanisms, in contrast to unicellular creatures. You know how many studies have been done since even before the mid-1950s on the responses of bacteria and then on yeast to their genetic responses to nutrients. And but it is only recently that it has been found that nutrients can directly affect the genetic expression in higher organisms. Metabolic changes modify genomic functioning through signal transduction pathways, transcription factors modifications, histone changes, microRNA modulations, among others. And this in turn, our metabolism is, as you well know, modified by nutrients. In turn, this reshaping of a genome operation modulates metabolic processes, leading to adaptations that optimize the nutritional state of the organism. The impairment of any step in the complex circuitries underlying these processes must lead to disease. Although we are just at the footsteps of their knowledge, even more on how they combine with abnormal nutrient intakes and unfit life styles. Their discovery will likely be one of the seminal developments of medicine in the 21st century. Efforts to identify components of the complex responses to dietary signals are becoming increasingly successful due to the advent of novel approaches, high-throughput techniques, the resulting data being interpreted by sophisticated data mining tools. As examples, I will present some parallel mechanisms that mediate genomic adaptations in the rat liver to dietary changes of glucose and of the vitamin biotin. The liver is able, as you well know, to store and to produce glucose. Absorption of carbohydrate in the diet is concomitant with increases in the concentration of liver substrates such as glucose, but also with changes in the concentrations of pancreatic hormones like insulin and glucagon. Differentiating the mechanisms by which hormones and substrates modulate gene transcription has been possible mainly by studies in isolated cells, such as primary culture hepatocytes or hepatoma cell lines. Two crucial transcription factors are the esterol regulatory element binding protein 1C, SRABP1C, and the carbohydrate response element binding protein, ChRABP. Of course, there are many more transcription factors, and what I'm going to do, because of didactic purposes and the short time of the presentation, is rather than to present in its known complexity, which probably is even less complex than the true complexity, I am going to simplify this to highlight the ideas rather to present a very comprehensive picture. The main effect of insulin in the liver is the induction of glucokinase. And here, the transcription factor SREBP1C plays a very important role, as we will see. Several lines of evidence suggest that glucose must then be metabolized to stimulate further gene transcription. Glucose 6-phosphate, the glucokinase product, is most likely the signal that activates the other transcription factor I am referring to, ChREBP. However, this is an issue that is not yet settled, and there are other possible mechanisms that have been proposed besides the production of glucose 6-phosphate, for example, through the pentose phosphate pathway. However, most of the evidence thus far points towards a critical role of glucose 6-phosphate. Now, anyway, whatever is the real culprit, what it is clear is that glucose metabolism is necessary through ChREBP to induce the transcription of other key enzymes, mainly related to lipogenesis, pyruvate kinase, acetyl-CoA carboxylase, fatty acid synthase, malic enzyme, and stearoyl-CoA desaturase. Thus increasing the amounts of these enzymes and activating the glycolytic and lipogenic pathways. This can be summarized in this scheme that I would like you to keep in mind, in your minds, because this is going to be a module of a more complex slide that I will present later on. But here is the insulin pathway that through a number of signaling proteins like PR3K, of these phosphoinositol 3-phosphate kinase, activate SREBP1C, which, as I have said, activates glucokinase. And then through glucose metabolism, seems that not longer working well. Anyway, through glucose metabolism, it activates ChREBP, that then induce lipogenesis gene transcription. On the other hand, it is known that SRABP1C represses the transcription of the gluconeogenic enzyme phosphanol pyruvate carboxykinase, PepsiK, and in this way the cell can get. So here you see that both the hormones and the substrates like glucose, how they interact in a higher organism. Is there I don't know if, oh, here it's working now. Maybe I wasn't pushing the button strong enough. In primary hepatocyte culture, insulin increases glucokinase in a biphasic form. You can see it here in terms of enzyme activity, and here is in terms of enzyme protein. The first phase is largely the effect of insulin through the transcription factor SRABP1C, as we have seen. But in the second phase, which occurs about 6 hours later, there is a further increase of glucokinase that also requires glucose metabolism. However, here an interesting thing happens. This second phase coincides with a sharp rise in cyclic GMP. If the breakdown of glucose, if one blocks the effect, the production of cyclic GMP, for example, by ODQ, which is an analog of guanylate cyclase, then this second phase is abolished. If the breakdown of glucose is blocked by the addition to the culture medium of N-acetyl glucosamine, a competitive inhibitor of hexose phosphorylation, the black circles, you see that there is no second phase. But if the medium is then supplemented with cyclic GMP, it is given in the form of an analog, 8-bromo cyclic GMP that can cross the cell membrane. Then you see that even in the absence of glucose metabolism, because of the effect of N-acetyl glucosamine, the second phase does indeed take place. It is well known that cyclic GMP is the product of soluble guanylate cyclase, and that its genetic effects are mediated by the cGMP-dependent protein kinase, also known as PKG. After PKG is phosphorylated by cyclic GMP, it then moves to the cell nucleus, where it produces a number of transcriptional effects. For example, it induces the expression of two important transcription factors, like C-FOS and JUN-B. Now I will move into the effects of biotin as we have been studying them in the last few years. We have been studying the effects of biotin starvation on carbon metabolism gene expression in the presence of abundant glucose. I'm going to present to you mainly the results that we have obtained studying rat liver, but we have done these studies also on the nematode, in the worm, C. elegans, and in the yeast Saccharomyces cerevisiae, and we have found basically the same kind of results in these three phylogenetically very distant species, eukaryotes. So when we have these organisms, and specifically the rat, when we starved it of biotin, but in the presence of abundant provision of glucose, what we found is an ATP deficit. But you can see this also in the other species. And you can see that when we restore the biotin deficiency by supplying the vitamin, then there is a recovery of the levels of ATP. Well, something similar happens to glucose utilization. Glucose utilization, we could, in yeast, we could measure glucose consumption, which of course it was very difficult to do it in the intact rat or even in the worm. And you can see how glucose consumption goes down in biotin deficiency, but normalizes when we restore the biotin. Now, here is the production of lactate in the case of the rat liver or the worm, or ethanol, which is the glucose product in the case of yeast, and you can see that there is diminished glucose utilization. Then we studied, with the aid of DNA microarrays, the transcriptome changes, and I'm just going to mention them. What we found was something very strange, paradoxical results, because although we were giving a lot of glucose to these organisms, we found a paradoxical diminished expression of genes for glycolysis and lipogenesis, and an augmented expression of genes for fatty acid oxidation and gluconeogenesis. So they were behaving as if they were not able to detect the glucose which was available at high amounts. These are the results, some results of real-time RT-PCR studies to quantitate some selected mRNAs, and you can see how in biotin deficiency they go down, as we saw in the microarray in the transcriptomic studies, and they go up when we give the vitamin, and the reverse occur with Pepsi K. This is just a control. This is holocarboxylase synthase, or biotin protein ligase. It may be pointed out that several of these this is something that I would like to mention, even if just very briefly. It may be pointed out that several of these findings are reminiscent of several ones observed in insulin resistance. Not all of them, but many of them. So you have high glucose available, like in a person with hyperglycemia, and instead of seeing glucose utilization, what you see is new glucose production and the use of fatty acids as metabolic fuels. We are also looking into the mechanisms underlying the above observations. The ATP deficit is sensed mainly, there are other sensors of course, but the main sensor of ATP is AMP kinase, AMPK, which I'm sure many of you are familiar with it. A family of crucial sensors and regulators of cellular response to metabolic stresses like glucose and energy deprivation. AMPK is activated by phosphorylation and allosteric modification by AMP. AMP goes up when ATP goes down, and produces extensive transcriptional changes through different signal transduction pathways. In the biotin-starved liver, AMPK was activated. So what you see here is using an antibody specific to phosphorylated AMPK, whereas here is an antibody to total AMPK. So you see an increase, which is this one, during biotin deficiency. But if we give biotin back, then it lowers to control levels. In the biotin-starved liver, then after, besides the activation of AMPK, several signal transduction pathways, and I'm just going to mention them, pathways: insulin, TOR, adiponectin, cytokine receptors, JAK-STAT, and several transcription factors and coactivators like SRABP1C that we have talked about before, and CHRABP, and also PGC1-alpha in the rat, and its homologues in C. elegans were modified. And these results were also reversed when, here you can see, CMPK, SRABPC, and TOR, PGC1-alpha, that some of you may know that it's so important for gluconeogenesis. These results were reversed when the missing biotin was replaced. We hypothesized that the above-mentioned findings may result, at least partially, from some of the functions of biotin as the prosthetic group of carboxylases. In particular, genomic effects of biotin starvation through anaplerosis impairment is appealing, since the main anaplerotic enzymes are the biotin-dependent pyruvate carboxylase, and to a lesser degree, also important, propionyl-CoA carboxylase. Based on our results, we proposed a heuristic model for a circuitry, heuristic in the sense that this is a model that now we need to test and we need to study experimentally. However, what you see here, it's our model, but here is biotin, which is missing, And therefore, this anaplerotic step catalyzed by pyruvate carboxylase is impaired, and we assume that there is impaired functioning of the TCA cycle with the consequence a decrease in ATP production. Green is diminished in relation to controls. Red is increased in relation to controls, and these are the ones that are red or green were actually measured by us. So here is, for example, you may remember the scheme I presented. The scheme I presented before. Here is the insulin pathway. This PI3K. This SREBP1C glucokinase. This CHREBP and the lipogenic enzymes. And this is the repression of gluconeogenesis, normally, which is now seen as REBP is low because of the effect on it of AMPK. Then we think that this could explain the increase of PGC1 alpha, at least partly the increase in gluconeogenesis. Now, we have not seen the metabolic flows themselves. We're studying them now through the use of glucose C13, but these are the transcripts of these genes.
2 more minutes.
And those transcripts, 2 minutes. Okay, I will go quickly. I'm going to jump through something that I was going to. Well, now this is important. In yeast, the above-mentioned changes were reversed when aspartate, the anaplerotic amino acid that is transaminated to oxaloacetate, was added to the culture. And here is, you have seen this before, but this new bar is aspartate. And you see that aspartate produces similar effects to biotin in biotin-starved, in biotin starvation. I'm going to go through this, which is another possible pathway, and we're also studying a crosstalk between these two pathways. But to finish, it is becoming evident that many common variants of very small effects mostly explain the genetic component of common diseases. Although thus far most gene disease associations have been seeked among proteins coding genes, it is likely that many genes for signaling and regulatory sequences have a substantial participation in the genetic component of multifactorial diseases. As an example, Keschwartz recently studied the association between single nucleotide polymorphisms in genes encoding AMPK and the risk of type 2 diabetes, and found that genetic variants of this pathway may exert a weak influence on the occurrence of this disease in the Japanese population. So in conclusion, the genetic study of variation of responses to nutrients, of the numerous components of regulatory circuitries, may hold the key to better understanding of common disorders. And so, as in the previous marvelous lecture by Professor Bouchard, what we see is that through the study of responses of genes to nutrients or to exercise, and through the studies of the genetic variation of the molecules that mediates these effects, this may possibly link into one the subjects of nutrigenetics and nutrigenomics. Thank you.